Great, so welcome everybody. I'm so glad you can all be here. I'm Kara Roselle, I'm the NOFA-MA Education Director, and I manage our soil health initiatives, and I'm here with Sue Schäufele, UMass Extension Vegetable Program Educator, and our guest presenters, Carol Glenister and Elson Sheilds. In a moment, Sue will give a full introduction of our guest presenters, but I just wanted to first take care of a little bit of housekeeping and share a little bit about the context for this event. So first of all, Sue is going to be posting a poll just now in the chat for you to fill out with just a few demographics questions for UMass's record keeping purposes. I see it there. Thank you, Sue. And so this event is actually part of a collaborative project funded by the Natural Resource Conservation Service, or NRCS's, Conservation Innovation Grants Program. This project involves NOFA-MA, Connecticut NOFA, and NOFA New Jersey, as well as UMass Extension, Rutgers Agriculture Experiment Station, and UConn Extension. And our project tracks soil health outcomes, pest pressure, and yield information on 9 farms across 3 states. Those farms were all selected because of their innovative approaches to reducing tillage in organic production systems. So as many of you on the phone might know, doing tillage reduction in organic systems is challenging, and we're trying to figure out the best ways to do that. So in a normal year, our extension providers would do field visits to help our farmers identify and track pest pressure. But this year, due to COVID, extension agents weren't really able to get out into the fields. So instead, we're using some of that funding to offer education around pest-related topics of the greatest interest to our participating farmers. And the topic of the role of beneficial nematodes in natural pest control came up as something of great interest to our farmer participants. So speaking of whom, I just wanted to recognize farmers Mike Rossweiler of North Slope Farm in New Jersey, Scott Morgan of Morganics Family Farm in New Jersey, and Alex Carpenter and Yoko Takamura of Osawaga Farm in Connecticut. They're all farmers in our CIG projects, and later during our agenda that you can see, they'll be kicking off our Q&A segment with just a few questions for our presenters. And after that, the moderators will present your questions to the presenters. But because of the amount of people we've had to register for this event, we won't be able to invite you to ask your questions live. So we just ask that you keep yourself on mute and drop your questions directly into the chat as they come up for you, and we'll organize those and do our best to get to all of them during our Q&A. And then finally, if you learned something today, please do consider making a donation to the UMass Vegetable Program and to your local NOFA chapter. I'll be dropping links into the chat for how you can do that. Okay, so at this point, without further ado, I will turn it over to Sue, who will introduce our speakers.
Thanks, Carol. I'm gonna stop this poll now and go back up to our agenda for a moment, and just thanks for the introduction, Carol. And I'm going to introduce our first speaker, who is Carol Glenister. She is the founder and president of IPM Labs in New York, where she works with farmers to get the most out of beneficial nematodes, as well as many other biocontrol agents. So many of you are probably familiar with Carol. And yeah, if you want to go ahead and share your slides, Carol, I'll stop here.
Okay.
And I did want to just make a quick, we would ask everyone to just try and keep yourself on mute. And if you're not speaking, you could turn your video off as well, just to help with bandwidth issues. We do have over 100 people logged in currently, so that would help. Great. Thanks, Carol.
Okay, hi everybody. Thank you for having me, Susan and Carrie. I'm gonna give you just about everything I know about nematodes today, briefly in an overview. And that was what Susan asked me to do. I'm not going to be presenting on the most recent nematode products, which I actually don't know anything about. But the ones that are used a lot, I will be talking about those. Okay, so this particular slide comes from a company called Becker Underwood, which sold itself to BASF a number of years ago now. And it was created by a woman named Julie Graish, who now works for BioWorks. But it's a beautiful slide, so I'm using it. So nematodes, they're almost magical. They're not magical. They have a characteristic that makes them extremely convenient, at least the standard nematodes, for commercialization. And that is that they have a stage called an infective juvenile stage. You can see that it's referred to right here, which can go dormant and it can be stored for a long time. That makes it commercially viable. That infective juvenile is the one that invades the insect through holes in the insect, like their spiracles or their mouth or their anus. And then they take over the blood system by inserting a bacteria into the blood, and they get the bacteria inside the insect anyway. And then the nematode, the bacteria multiplies inside of the insect and takes over the insect and kills it. And then the nematodes start to multiply and feed on the bacteria. And then they produce 2 or 3 generations, and then new infective juveniles come out. And those juveniles will be the ones that will be harvested by the company that was going to sell them and then sold for controlled pests. So that is a particular characteristic that makes it possible to do this commercially. We also have Heterorhabditis, another genus, which doesn't store so well. So this is a particular characteristic of the Steiner nematodes. They do store a bit, but not as well as the Steiner nematodes. So as growers, you want to know how to keep nematodes happy. And it's important to know how to kill them so that you don't do that. So one of the things that is really important is that you don't subject them to sunlight, because sunlight has a lot of ultraviolet light. Ultraviolet light can kill them within about 30 minutes. So what people like to do is apply them at dusk or apply them in the rain or on a cloudy day. Also, nematodes drown easily in water, and we're usually spraying them out. So you don't want to leave them in water for more than 2 hours, because they're going to run out of oxygen and they're going to die. So if you do have them in deep water, you can prolong their life by oxygenating the water with a bubbler. So that's another way to go about it. If they're going to move around in the soil, they have to have a film of water to do that. So one of the things that we do is water before and water afterwards in order to give them that film of water to travel and find their insect host. Now, if it's not convenient to water before and after, it can be very convenient to apply in the rain. So if you can hold off until you're going to have a rain event and make sure that it is raining while you're applying, that's fantastic too. So nematodes have to be able to wiggle to find their host. They have to be able to invade their host, and they can't do that if it's too cold. Some species function okay above 50 degrees. Steiner Nematodes is one of those. Another species, Heterorhabditis bacteriophora, and Steiner Nematodes, which I'll review in a few minutes, they don't work below 60 degrees Fahrenheit. So I consider the heteros and the Carpocapsae to be more of a warm nematode, and the Steiner Nematodes to be more cold tolerant. It doesn't mean that they're not going to tolerate ice. It's just that they can't function. You can actually ship the nematodes on ice. It's not going to hurt them, but you cannot expect them to actively work when it's not warm enough. Another thing that's really important, and it's the reason that they were given carte blanche by the Federal Register, by Congress, to not require registration with the Environmental Protection Agency, is that they don't do well above 85 degrees at all. They can't, infect human because we're too warm. They don't do well in mammals because they're too warm. And so if you leave them in water above 85, or even if they're in soil that's too warm, they're going to die. One of my favorite things in a show and tell is to take some nematodes and heat up, get some hot water for tea, and put the tea water on those nematodes, and then show everybody the dead nematodes. They die very quickly. So I can have dead nematodes and live nematodes next to each other just by having poured some hot water on them. So you can apply them a lot of different ways. The main thing is to distribute them over the soil and get them in with water. Some people actually have apply a granular formulation, which they water in afterwards. It's not easy to distribute. So almost everybody is spraying one way or another. You can spray with a watering can. It doesn't have to be a sprayer, but there's big commercial sprayers that are used. Some people put them through their watering system. It's really important not to have fine screens on. So if you're using a pump sprayer, you have to take the fine screen off that is in the nozzle and possibly look for other fine screens. It's good to make the nozzle aperture coarse, and above 300 pounds per square inch pump pressure is going to start mutilating the nematodes. So don't use high pressure.
Carol, do you mind if I interrupt you quickly to ask a question?
Sure.
There's been a question, and I'm sure other people have this, is can you apply them through drip lines, or do they get gummed up in there?
Yes, people do apply them through drip lines. I would not say that you can do it in general, though. You have to test for your own system to see if they're going to come through. Another thing about nematodes is they are heavier than water. So if they're going to settle out in any place along the way in the drip line, if there's enough turbulence, they'll come through. But you have to make sure they're going to come through on your farthest nozzle. And I'll show you a little bit about how you might be able to do that in one of the last slides. So it's possible. The other thing I don't like about drip lines is that the drip line is going to deliver the nematodes right where the drip comes out. It's not going to disperse them. They're not going to be well dispersed. So sure, if you've got water uniformly throughout the profile that you need them in, maybe the nematodes can get to where you need them to be, but I wouldn't necessarily trust that. So you can, but with caution.
Okay, great, thank you.
You're welcome. This is another nice slide from Julie Graish. Here we've got nematodes coming through a broom waterer in a big greenhouse. So here's a backpack sprayer. This is Roger McGaughey from Pioneer Gardens, who is operating one of those big sprayers that you saw. Another commercial sprayer. And this is a dosatron, which is taking nematodes. This is a concentrate with the nematodes out of the bucket down here, and it's metering that into a watering system. So a dosatron can be very convenient in the greenhouse, but many times you're going to be outside, so you're probably going to need some kind of a sprayer outside. Most pesticides, organic pesticides, are going to be compatible with nematodes. I wouldn't worry about that very much unless you know them to be nematicides. If they kill nematodes, if they're used for nematode control, I'd stay away from them. I can't think of one that is harmful. Maybe Elson will have something to say about that, but I don't know of any. So what we're using now is nematodes for these pests: black vine weevils, fungus gnats in mostly greenhouses, grubs in lawns and in vegetable production, shore flies, scales on foliage plants, strawberry root weevil, which is very similar to black vine weevil, and thrips. These are commercial uses. And I have guess I didn't spell this out in advance. This is Steinernema feltii. This is Heterorhabditis bacteriophora. We use those for black vine weevil. The Steinernema would be used for the cold time of the year, and the heteros for the warm time of the year. It's important to attack the stage of the insect that is vulnerable. Nematodes are not going to last forever, so you need to put them down when the vulnerable stage is there. And in the case of black vine weevil, the vulnerable stage is larger larvae. It's true also for strawberry root weevil and grubs. The larger larvae are going to be just before pupation in the spring and then in August. Since heteros like warm weather, they don't like cold weather, I would say that'd be August for the heteros because you have large larvae in September, October, November, but it's too cold. So you've got to get those nematodes down when it's still warm. And then in the spring, you've got to get the Steinernema down before the black vine weevil pupates. If you're seeing pupae, it's not going to work very well. It works better against the larvae. For fungus gnats, we use Steinernema feltii, and that is against the larvae. So the best time to apply is when you plant in the potting mix where the fungus gnats all lay their eggs. The potting mix will be protected for many weeks, and as the larvae hatch, and the larvae will be killed by the nematodes. For lawn grubs, that would include many, many different kinds of grubs, including Japanese beetle grubs, which people are very interested in controlling, and the Asiatic larvae. Steinernema feltii for the spring before it gets warm, and when the grubs are large, and heteros in August. There are many kinds of grubs, and I prefer the heteros myself, but people don't usually want to wait until August to make that application because they want to do something right now. And the application in August is going to be control for next year. It's not control for this year so much because the damage is already being done this year. But the heteros can actually drill through the exoskeleton of the grubs, whereas Steinernema feltii cannot. And grubs have these resistances to nematodes. So this is the strongest nematode that I'm working with right now that can control grubs. I think there are a few more coming down the pipeline. I am not familiar with them. You want to keep track. There's one which I can't even name because I didn't have time to figure it out, which apparently is very cold active too. For shore flies, that would be a greenhouse pest where there's a lot of algae. Steinernema carpocapsae works quite well. We are using feltii for scale control on foliage plants. Some people swear by it. No scientists have really supported. They haven't even tested it because there's too many scales and nobody wants to pay for the research. However, I can tell you that long ago, there was some research on whitefly control with nematodes, and it worked. It just didn't work as well as some of the other things we have. And whitefly immatures are scale-like, so it makes sense to me. Strawberry root weevil, heteros in August, and that's for your next year's strawberries. It's on older strawberry fields. And then for thrips, Steinernema feltii is being used quite a lot. It's usually applied weekly, and I'll talk more about that later. How are we doing for time, Susan? I can't really see.
Let's see, we have, I think, 6 or so minutes.
Okay.
But we have a lot of time built in for question and answer, so there's some flexibility there.
Okay, so the rates range a lot. They're commonly anywhere from 25 to 50 million, but you're going to see a rate here shortly that wasn't in the 90 million range. They have different reasons, but that's the rates that are usually coming up, and different producers recommend different rates. And growers do whatever they want. So here's one that would be very interesting, I think, to cabbage growers. Cabbage maggot control. can be done with Steinernema feltii. And in one case, a conventional farmer, Barber Farms in New York State, has been controlling cabbage maggot since 1995 with Steinernema feltii. And before that, they had quit using pesticides and they were sustaining 50% damage by not doing anything. And before they did nothing, they were using diazinon and got clean cabbage. So their rates, and here's a 90,000 per square foot rate, was to drench the plugs, the little cabbage seedlings, with nematodes, watered on with a watering can before they planted them out. And then when they planted them out, they had some dribbling in with the transplant water from their big tank, their 500-gallon transplant water tank, and they only put 25 million in with that, but it was going in right where the cabbage plant was. And it makes sense to me. If you have a pest in a certain place, put the nematodes there. Don't put them everywhere. And you can even use that for figuring your per square foot rates. You know, you might not do it for the whole acre, you might do it for the quarter of an acre. Do your calculations for the quarter of an acre where the plants are. So seed corn maggot, people really want seed corn maggot control. Some people have told me they have great control, but I can't make the recommendation because you can apply nematodes for seed corn maggot and all the seed corn maggots will go away, but they do every year because it's generational. The generation's there and then it disappears. And I don't really know if they have disappeared because of the nematodes or if they disappeared just because of the generations of the seed corn maggot. So we definitely need research here. Seed corn maggot is a horrible pest. It destroys lots of organic crops. And it would be really great if nematodes really did that work. I just don't know. And I already described the grub control, but I do want to say something about grassy areas and grubs. New vineyards are very, very, very prone to a Japanese beetle if they till up the whole vineyard and then plant new grass strips in, because the new grass strips are the best thing Japanese beetle ever saw. And they survive and thrive so well because the whole community that kills Japanese beetle grubs was destroyed in the tillage. So if you can avoid tilling a whole area and leave some of the natural enemies in the site, like in a vineyard, you can avoid that whole upsurge of Japanese beetle just by leaving the turf, some turf there. Here's another thing. I don't know quite how you could use it, but I know conventional growers use it. They compromise a grub's immune system with a low rate of pesticide when they treat with the nematodes. And I don't know if that would work with neem or not, but it's something to, if there's an organic pesticide and you really have a tough pest, perhaps wireworms, you might be able to enhance the control by doing both the nematodes and a light rate of pesticide. I already discussed this. This shows you, sorry about the image, I didn't have time to find a better one, but this is nematode killed pupae, nematode killed grub, next to healthy ones. And so they're dark and brown. In the case of the heteros, they have a reddish tint, but they look hurt, I guess. Here's some nematodes coming out of a black vineweevil pupa, and that would be the infective stage coming out. This is a question, and I tried to give you a bit of an answer there with the not disturbing turf. This is the question I'm always asking, and I'm sure any natural grower wants to know the same thing. There's many tricks. I can't address it today, though. Thrips control is done quite a bit with nematodes in the greenhouse. It's a weekly treatment. Scientists still dispute that they're being killed up on the foliage, and they say they're just being killed in growing medium, but we know that they're dying. really well. So these are all Western flower thrips. Nymphs biocontrols and nematodes are one of them. This guy has been a pioneer. His name is Roger McGawkey. At that time, he was at Michael's greenhouse of using nematodes in foliage, and seeing a greenhouse full of flowers in July and not having any thrips anywhere convinced me that he had something. He does a weekly treatment. Here's one of his workers spraying hanging baskets, which I wouldn't do because I don't think you can get good coverage, but they seem to like it. And this shows that a wetting agent is required to keep the water from balling up on the foliage. In this case, for thrips control, the water has to flatten out so the nematodes have more area to cover. So when you get your shipment of nematodes, if you're buying nematodes, make sure that it's cool. Check it to look for the dates on the packages if there are, to make sure they're not old. And you can check to see if the nematodes are active by the following. This is another great slide from Julie Graish. Here, this this demonstrator has, where's my, right here, there it is. That's a hand lens. Sorry. That's a hand lens in the hand there. And you take a little sample of nematodes. It could just be a paper clip that you dip into the nematodes and put into a droplet of water. Very small sample. If you get too many nematodes, you can't see anything because there's too many. And then you put a flashlight, see the flashlight right down here underneath, and you look down through the hand lens. You have to get your eyes really close to the hand lens to see this. And you look to see if there's any movement of the nematodes. If they're wiggly, if they're curved, they're alive. If they're dead, they're straight like toothpicks. If you see any wiggling at all, they have to be alive. The toothpick ones, they could also be too cold. So you could warm them with your hands. You could put a few grains of salt or grains of fertilizer in to get them activated. But the wiggling ones are alive for sure. Storage, when you get them, put them in the refrigerator until you use them. The Steinernema can last for up to 6 weeks in the fridge. The heteros, I wouldn't keep in the fridge for more than 2 weeks. And fundamentals of successful biocontrol, but I think they're fundamentals of a lot of pest control. You got to use the right biocontrol agents, that's BCAs, for the pests identified. And you need to have the biocontrols constantly exerting pressure, and you need to use compatible pesticides if you're going to use them. These are my acknowledgements. Thank you to Julie for her fine work. And thank you all. Any questions?
Thanks a lot, Carol. That was so much information. It's great. And there were a lot of questions. We wanted to involve some of the farmer collaborators that are part of this larger grant project to facilitate some of the Q&A. And my apologies, Carol, are we doing that now, or should I ask the questions of Carol and we'll do the farmer questions later?
I think the farmers are going to kick off the panel Q&A for both presenters, and we just have a little time for specific questions for Carol before we move on to our speaker. And I just sent you like application questions, and then there was questions. I'll send those separately.
Yeah, so there were a lot of questions that came in, Carol, and hopefully can stay for the end where we could have maybe more of a longer discussion. Some of the particular questions were about certain pests, questions of would the nematodes work in which species for slugs, flea beetles, and onion maggots. Those were ones that came up that you didn't mention yet.
Okay, no problem. Slugs, no. There is a nematode that was created, or a product that was found in England that has commercialized, and they have just found one in the US recently. I am not aware that there's a commercial program yet. So not yet for slugs, sorry. For onion maggot, I wouldn't make the recommendation for onion maggot, and I am more certain about that than I am about the seed corn maggot. And the reason is that the onion maggot eggs are laid in such a way that the onion maggot can get into the onion very quickly. So the nematodes aren't going to get in there. However, for onion thrips in a high tunnel in Minnesota, it's been done for many, many years. It does a great job in controlling onion thrips out inside. It hasn't been tested outside. So onion maggot, what was the last one?
Flea beetle.
Oh, yeah. So flea beetles are coming.
You can save that for the end if that's better.
If they were to kill flea beetle larvae, and they can, they would be killing flea beetle larvae on where you put the nematodes. So you might be able to pull something off if you created a trap crop and then you treated around the base of the trap crop in order to kill the flea beetle larvae at the trap crop site. But to protect your crop from flea beetles that are coming from all over the place, all your mustard weeds, everywhere. I don't make that. I never encourage people to believe that.
Okay, thanks for that. And again, we'll have more time for questions and answers at the end. Elson, are you able to share your slides now?
Yes, I can.
Super. Well, thanks again, Carol, and welcome to our next speaker, Dr. Elson Sheilds from Cornell University, Department of Entomology. Dr. Sheilds has been working since the early 90s on using sort of a different approach, native persistent nematodes for pest control. And I'll let him take it away. Thank you.
Well, thank you. So Carol has done a good job of talking about the commercial nematodes, and I'm going to take you in a completely different direction. And so we're going to talk about nematodes in more of a classical biocontrol. And this was developed in the field, in field crops in New York, and now it's spun off to where there's some uses in organics. So we want to talk about that. But the first thing I want to do is back up a little bit and try and pre-answer some questions that you'll all have. There's a lot of confusion when we get to talking about nematodes because we have free-living nematodes, we have plant parasitic nematodes, and we have animal parasitic nematodes. And there's a fair amount of confusion that if we release entomopathogenic, will they attack plants? So I have a list here of some famous nematodes that are specialists that we are familiar with. So soybean cyst nematode is a nematode, attacks only soybeans, very specialized. Root knot nematodes, a plant parasitic nematode that you all may be familiar with. It's a broad spectrum of vegetables. So those are plant parasitic. Now we have some nematodes that are vectored by mosquitoes. Elephantiasis is in the tropics. It plugs up your lymph system. Most of us have dogs that we treat for heartworm. That is a nematode that's transmitted by mosquitoes, very specialized. And there are nematodes that like our GI tract, so what we are talking about is a special nematode, entomopathogenic. I tend to use biocontrol nematodes. I stuttered as a kid, and sometimes if you say entomopathogenic too many times, it kind of gets tongue-tied. So I use them interchangeably. These are, as Carol mentioned, an obligate parasite of insects. They're naturally occurring in the soil. You have entomopathogenic nematodes on your farm. They kill their host and they reproduce within their cadaver. So that raises the question, a couple of questions. First of all, where we look, we find entomopathogenic nematodes. They're virtually in every soil habitat where in the temperate regions. So the question is, why don't they work? The native nematodes in your fields are unlikely to be adapted to agriculture and agricultural pests. Therefore, they're not providing sufficient pest control of soil insects. And so My whole goal has been to see if we can figure out why those native nematodes aren't working and see if we can adapt them to agriculture. So the first question is, and some of this will support what some of Carol told you, is there a soil profile preference between the species? And the answer is yes. Carpocapsae is only happy in the top 2 inches of soil. You can water them down 6-8 inches, they return to the top 2 inches. They are the most common nematode naturally in New York soils, and virtually we can find them in any field. Sanonema feltii goes down 8 inches. It's both an ambush and a cruiser, and we find it isolated pockets in New York. For instance, one of the hot spots for feltii happens to be the Hudson Valley. H. bacteophora, Heterobidites bacteophora, is in isolated pockets around New York, primarily in the sandier soils. So those species that Carol talked about are actually here in New York in isolated pockets or widespread in the case of Carpocapsae. So can we mix the species to be effective? You know, if you look back at this, you think, well, maybe Carpocapsae rules the top 2 inches. Can I mix it with some of the other species? And the answer is yes. We've shown that if you mix Carpocapsae with feltii, they do a really good job in the agricultural layer, and that's the best combination for the heavier soils, the clay loams to the loam soils. If you have sandier soils, we find that feltii and bacteophora work better. So this is a fair amount of research has gone into this. How do they persist? I mean, the commercial nematodes are good for a week, 2 weeks, 2 months, but they're gone by winter. Well, your wild nematodes have phased infectivity. So not all the IJs in the soil are infective at the same point in time. It's genetically based, and if you take the wild New York nematode and continually rear it, they'll lose their ability to persist. So the minute you take these and commercially start rearing them, you lose the ability to persist in the environment. This is an example of 2 of the New York strains, and what you have is feltii and Heterobidites. 363 days later, at room temperature, you can see we are still killing Glary. Now, feltii is now out to 3 years. It has been at room temperature for 3 years, no recycling, and we are still killing. So that is persistence. So let's draw some contrast between what Carol talked about and what I'm talking about. So on the left, the commercial strains are easily purchased. They are used in a more biopesticide approach. They're present from 7 to 90 days. They're much more virulent because they're 100% effective. If they're alive, they're infective. They've lost the ability to persist, and they tend to be faster acting. The native strains of the same species will persist for years from a single application. They're less virulent, but then do they need to be because they're there year in and year out. Only a portion of the population is infective, which is why they persist, and then they're a little slower acting. So the 3 isolates that we have experimented with, and you'll recognize the name, Sanonema carpocapsae, we call it New York 01, feltii, New York 04, and Heterobidites bacteophora Oswego are the 3 strains that we isolated in New York in the 1990s, and we go back and re-isolate them every 2 years. So how did we stumble on? And I literally say stumble on to this technology because what you'll find is the Sheilds lab is the only one in the United States that has been doing this, and everybody thought I'd been smoking crack in the alleys of New York while we started this because we were going in a completely different direction. Well, we are blessed or cursed with alfalfa snout beetle. This is an Odorhynchus species that is 1 species away from black vine weevil and strawberry root weevil, and it kills alfalfa in a single year, and it's devastating to the dairy industry, and it cannot be controlled by insecticide. The entire North American infestation is what you see. 9 counties in New York, and a little bit of Canada. So I was given the task early in my career of solving this problem, and being a smug young assistant professor, I said, yeah, 5 years, I'll get it done. Well, 30 years later, we have achieved it. And you see very early on, snout beetle is very sensitive to nematodes. Here's the nematode treated plot, and you can see where alfalfa has been killed out. And this is an issue of persistence. So here's an alfalfa field that's 6 years, you know, and we have 30% of the soil core showing activity for nematodes 6 years after it was inoculated. And this is a combination of Feltii and Carpocapsae. So, you know, that's interesting. If you look at different fields, here is one that obviously had a lot of insects in it when we applied it, and we're jumping up to 3 out of every 4 soil samples are positive, and it drops down after the infestation is gone, but it maintains itself around 25 or 30%. Here's one that's 3 years after the initial field inoculation. Single inoculation, suddenly we had an invasion of insects, and we're talking about 100% of the soil cores showing nematodes. And I have one more as background. Here's a field that 5 years after application, here's 4 years, you can see that the rates have climbed up to 1 out of every 2 soil samples, and 5 years later, it's 100% of the soil samples, obviously responding to an insect invasion, and it was applied 5 years before. Okay, so snout beetle is a serious problem. Farmers are desperate. We have more than 28,000 acres now that have been inoculated with persistent biocontrol nematodes on more than 130 farms in northern New York. In those fields, snout beetle is disappearing. Stand life has increased from 4 to 6 years. So having solved this problem, I decided to look a little more broadly and see if there are other applications. And so black vine weevil, we had 10 acres of strawberries. A farmer came and asked us, he was losing 10 acres of strawberries to black vine weevil. He'd been spraying insecticides and it wasn't helping, so he was open to us experimenting on his farm. And so I have a couple of more complicated graphs for you. Take a minute to explain them. So this is the number of plants that were infested with black vine weevil in the spring of 2013. And you see almost every other plant was infested with black vine weevil. Since black vine weevil is most susceptible in the fall, we applied our nematodes in the fall. And the following spring, we didn't seem like we had any impact. Well, the farmer was ready to shoot me, but I convinced him it takes time. The following spring, we sampled, and you can see that nematodes became active to where within a month, we went from 1 out of 2 plants to 1 out of 4 plants to 15% of the plants actually infested with black vine weevil. And superimposed over the top, you can see the increase in terms of nematodes. Now, that was spring of 15. You notice 16, there's no black vine weevil. 17, there's no black vine weevil. You just see a little bit, and our nematodes are persistent. A single application basically saved his 10 acres of strawberries. Now, if you look at another metric, which is actually more telling, this is the number of black vine weevil larvae found per plant. You see, in the spring of 14, we had 2 larvae on average per plant. But when we started seeing impact, the first impact we saw was a reduction of the larvae per plant. So we got down to less than 1. So that was black vine weevil. Single inoculation in 2013 reduced the population to non-detect 2 years later. Strawberries returned to full production, and we have not detected any black vine weevil populations. And we quit looking at 2019. Wireworms. I heard Carol talk about wireworms. Well, at the Farm Hub in the Hudson Valley, we tried persistent nematodes on wireworms. So we set up 4-row plots by 50 feet. We treated untreated check, Carpocapsae by Feltii by Heterobidites. We inoculated the plots in 17, and then the first data will show you October of 17. So here we are applying ATV sprayer, basically streaming the nematodes on with fertilizer stream nozzles. You can see it's a sea of hosts for wireworm. This is ryegrass. Setting up our plots. The first question is, on these sandy soils, will we see persistence? And the answer is yes. So here's the percent of soil samples that are positive. You see here we're in this magical 25 to 35% of the soil cores being positive. So this is Feltii, this is Carpocapsae. If we look at the other mix of Feltii and Heterobidites, you see the similar things. It's a little deceiving, these low levels of Heterobidites, because it's a cruising nematode. It's kind of like trying to chase down something that's moving all the time. But from 17, the application, to 2019, we have long-term persistence. Well, is this killing wireworm? That's what we really want to talk about. So if we look at the untreated checks, the fall of 17, you see that both the outer two rows and the inner two rows had 73 mines per, and I don't remember the quantity, 73 mines, probably in the entire sample size. With Feltii and Heterobidites, that dropped down to 30 and 41. So you have almost a 50% reduction in the feeding damage with Feltii Heterobidites of wireworm. With Carpocapsae Feltii, you have a smaller reduction of it. So obviously, Feltii and Heterobidites did a better job. So that was in year 1. In year 2, we had taken out the wireworm life cycle is a lot of the adults had left. So, but we still got a significant reduction between 27 feeding wounds and 14 from Feltii Heterobidites. Not significant with Carpocapsae Feltii. So we applied nematodes, they persisted, and we got reduction. Now we saw no wireworm damage on sweet potatoes in 2019. White potatoes were planted in 2020. There was no damage, and we have persistence through the 2020 growing season from a single application made in the spring, May of 2017. Other insects. You all talked about seed corn maggot. Well, I have money to start research in 2021 about persistent nematodes in seed corn maggot and cabbage maggot, by the way. And to answer the question about onion maggot, Brian Nault actually has an ongoing project with persistent nematodes and onion maggot. They just finished year 1. Flea beetles is a problem, as Carol mentioned, because of the movement adults, which cause the damage from the field edges in the spring. And to really be active, you would need to treat with persistent nematodes all of the outlying areas where you have the wild host. So it is possible with persistence to actually treat the overwintering areas and in addition to the field. That's the only way I can see to tackle it. With commercials that don't persist, that's not a viable strategy. But with persistent ones, we get tired of looking after 10 years of persistence. And so that's a native New York. White grubs. Well, the native white grubs are very susceptible. The Philophaga white grubs, multi-year, are very susceptible. Annual white grubs are a bit more of a challenge. Certainly European chafer is almost immune to nematodes and everything like that. For those of you that raise potatoes, white potatoes, I just finished a project which show that if you treat the population, you can actually get a reduction of Colorado potato beetle damage. And I have a current research project ongoing with Brian Nault to try that out on a larger scale. This picture was sent to me by a farmer. And what he did, and farmers are very creative souls. He applied nematodes to half the field, and then he planted his crop perpendicular to that. So in the same row, you would have a nematode treated portion and an untreated portion, and it speaks for itself. You can see the reduction of damage. And we've been trying to figure out kind of, is that wireworm damage, is it flea beetle damage, what it is. It really doesn't matter. It's quite telling. So EPN application, Carol talked a little bit about it. The persistent nematodes are reared on wax moth larvae. 2 weeks later, there's this goo that needs to be washed and separated with screens. You can see the screening, and filtered a second time, and then they can be applied, as Carol mentioned, in any commercial sprayer, watering can, etc. In the commercial market, we have had applicators apply them with 120-foot booms. So some of the big commercial applicators in northern New York against snout beetle are applying 300-400 acres a night with huge sprayers that have 120-foot booms using this method. For the smaller person, you know, you can use the smaller sprayer here, and you just take out the screens, the nozzles. And in fact, if you have a short boom, you can just take all the guts out of the nozzle body. One of our farmers in snout beetle country had no sprayers, so he just drilled a hole in a piece of PVC pipe, put it on his ATV and gravity feed, and got excellent inoculation of nematodes. What does it cost? Well, our rates are much lower, 50 million to 100 million per acre, not 10,000 square feet. It's a single application. It's a 50-50 mix of two species. If you're less than 5 acres, that comes out to $70 to $140 per acre. Single application, and not an annual application. If you're more than 5 acres, and this is just basically the effort that it takes to do them and deliver them, it's $50 to $100 an acre. The difference between 50 million per acre and 100 million is how impatient you are as a farmer for instant control. 50 million will do fine. It'll take a year. We typically see full activity in year 2 because we have phased infectivity, and it takes a while for them to get established in soil profile. We get some activity in year 1, full activity in year 2. So just to summarize, the natural occurring EPNs are poorly matched to agricultural soil pests once the land is converted to agriculture. You know, most of our farms are white pine forests. The agricultural or the active layer in white pine forest is only 2-3 inches. If you native adapted EPNs match to agriculture, persist in the fields for multiple growing seasons from a single application. And a single inoculative application of the correct mix of adapted persistent EPNs enhances the control of our soil insects for multiple growing seasons. And this is proven research. We are actively working on corn rootworm in corn and commercial agriculture, and I have plots all over the United States, including applications through center pivot systems in Texas and New Mexico. So this is more than just a little effort. So have the local organic farmers bought into it? There are 14 organic farms in New York that have applied, 1 in Vermont, and we've shipped to 3 organic farms in Canada. So what's the source? You know, as you notice, I'm old, I'm gray haired, and I'm sliding fast into the grave. So we've helped Mary DeBeer set up a business. She's primarily working with the dairy farmers in northern New York, but is expanding her business. And there are some safeguards one needs to do to make sure you don't lose the persistence. And if people are interested, we can help them. I mean, there's a big hole in supplying the organic farm. So if people are interested, we can teach you how to do this. Or you can contact us directly, and we can supply the nematodes. So with that, that's my presentation. I'm open to any questions.
Excellent. That's awesome. Thank you so much, Elson. In a unlikely or uncommonly, we are way ahead of time, That's my fault for getting ahead of schedule earlier on, but it's good. We'll have plenty of time for all of your questions for Elson and Carol. And we do have some farmers on the line who are going to be facilitating a discussion, unless you want me to ask some of the more specific questions at this time, Carol.
Yeah, I was thinking, why don't we invite Mike, Yoko, and Alex, and Scott to come off of mute and turn on your videos. Hey guys. And what we'll do, I think, is we can ask them to sort of kick off our questions by asking a few live questions so we can be sort of interactive. And then we can get to all of the great questions that the audience has been putting in the chat. And if there are any questions that come up that sort of cross between kind of farming practices and soil health and nematodes, maybe there could be a little bit of discussion if it happens organically. So great. So can you guys, maybe you guys want to just take turns coming off mute. I think what I will do is invite y'all in order of, oh, Mike can't unmute.
I can help with that, I think.
Awesome. So we have Mike Rossweiler from North Slope Farm in New Jersey, Scott Morgan from Organics Family Farm in New Jersey, and it looks like Yoko, you're here with that, Alex, from
Yes.
Farm. Great. So I think, Yoko, you're the only one who's off mute so far, so why don't you go ahead and ask your question first, and then we'll kind of bounce around.
Okay, great. Thank you so much for the presentations. It was really, really informative. I've got a few questions. I don't know how much time we have for these, but my first question, Carol might have touched on a little bit, but should we be worried about any imbalances in the soil that nematodes might create, competition with other soil species? Is there such thing as over application of these nematodes, and just overall negative effects of these nematodes, if there are any?
I've always been interested in that question. I think that people are going to limit naturally their expense, so they're not going to pay for an awful number. But there was some observations many years ago that if you over-applied nematodes, you could actually end up killing plant parasitic nematodes. And the reason was that the environment seemed to be backlashing against the nematodes by creating all these nematode feeders, and the nematode feeders were going against other nematodes in the soil. So that created an interesting thing, but the problem was it just cost, you had to use about 4 times the recommended rate, and 4 times the recommended rate is very expensive. So it doesn't really make sense to do it that way. Elson, do you have any comments on over application?
Well, I think that what you mentioned is there is a group of fungi that actually trap nematodes, and so if you get a high rate of nematodes, you'll promote the fungi that actually have a loop and actually trap the nematodes. I mean, a biopesticide approach or using commercials, you tend to over apply, but they don't last very long. So you're talking at a momentary pulse of lots of nematodes, then they die off. With the persistent nematodes, they seem to come to a, excuse me, a balance. And we cannot cheat and apply too many, it doesn't seem to help. So there is not a rate response with persistent nematodes. So if you apply 4 times more and you come back and sample, you still come to the same level as if you applied 1/4 of them. And so there's kind of 2 different philosophies here. And we think that we've not seen a problem with our persistence ones. And I think they evolved in New York. I mean, come on, you know, they're going to stay there in New York. We're not introducing anything that's foreign. What we have done, so agriculture is artificial, right? We chopped down white pine forest. We created agriculture because we like to eat. Well, I think that all we have done is found a mix of native nematodes that work in agriculture. So, you know, we're not introducing anything that unusual.
Okay, thank you.
Related to that, could I throw in the question just about what's the process of adapting that native nematode to serve an control in an agricultural crop? I mean, sort of, for me, it goes back to the question of, what is the process that you identify the beneficial nematode?
Okay. So there is no process. We know where we collected the original ones in the 1990s. We let Mother Nature keep them adapted to New York and Massachusetts and New Jersey. We're not that different really in soil environment. And we go back every 2 years and restart our host colonies to make sure that we haven't introduced something artificial to impact their ability to persist. What we have done is mix 2 species so we have coverage of the agricultural profile. So Carpocapsae is extremely common, but it doesn't do the job because the soil insects are below the Carpocapsae layer, right? They rule the Carpocapsae layer. We add Feltii to Carpocapsae in the heavier soils to actually get a profile coverage. But it's the same native New York nematode that has been adapted. We go back every 2 years. So we have identified the source in nature, and we go back and every 2 years make sure that we retain what's there.
Are you going back with waxworms, or are you using weevils? What are you using to capture the nematodes?
We use waxworms.
That's easy.
It is, and so we just bring in soil samples and and re-isolate and start from there.
I'm a little confused. So in the nematodes that you have identified that are effective, are they having sex in effect with the native nematodes and creating a new species?
No. We are going out. So we originally found Heterobidites Bacterophora Oswego in Oswego County, hence the name. We know the field we first identified it in, collected it. Every 2 years, we go back to that field and re-isolate it. So we're going back to the original populations. We know where we found the original Feltii. We have since found a better source of Feltii in the Hudson Valley. So we go back to those fields and we re-isolate every 2 years. That's all we do. And then we build them up and then they become a commercial. What Mary DeBeer does, and she's completely independent, is she went out and created her own repository in 2 or 3 yards of Heterobidites and Feltii, and she's up at Malone. And they are being adapted to that chill condition. And then she goes back out and takes soil samples. And so we use Mother Nature to make sure that they don't become too artificial. That answer your question, Mike?
Unfortunately, I mean, yes and no. I mean, I guess what I'm wondering about as an independent farmer, we've been involved in this grant, this conservation innovation grant, where we're looking at doing reduced tillage. And one of our farmers who may be on this call, and I'll shut up so they can talk about their experience, but has been experimenting with using nematodes to see if it's effective. And I can't help but wonder, when we start talking about the native nematodes, if there's a way of figuring out for my farm here what in that soil biology would be beneficial, that would be unique to my setting, that I could try to foster that population so that I could see natural, persistent benefit.
Well, first of all, if we were to go sample your farm intensively and bioassay the soil samples, we would find probably a single species of one of those nematodes on your farm. Where are you located in Jersey?
Central Western New Jersey.
So what soil type do you have?
It's a Lehigh, sandy, silty loam.
Silty loam. So So that used to be white pine forest, and I bet you that we would find carpocapsia. Carpocapsia is only happy down 2 inches. That's why you're not seeing an impact. What we do is we come in and we would add Feltii, which is also native to the Northeast. We could probably find it given enough money in New Jersey, but they seem to be adaptive. I mean, believe it or not, the Northern New York Feltii adapts to Roswell, New Mexico under irrigation, which is Roswell's like on the edge of hell. You know, I mean, it's hot, it's dry, and they've adapted down there. Fine. So they're pretty adaptive. But all we do is take what you have and add a second species so we get coverage of the whole profile. So you can pick them up from New York that are native, and they'll adapt to New Jersey, probably, or you can spend a lot of money and sample all of New Jersey and find your own Feltii, you know.
Just to build off that question and just to claim a little moderator's privilege here. So if I can follow up on what you just said about, you know, the native nematode you're mentioning is happy down to 2 inches. I think one of the questions that we're interested in as part of this project for tillage reduction in organic systems is, what are the specific farming practices, if you're aware of any, that can, you know, foster more nematodes in our soil? And does that include minimizing shallow tillage? Are nematodes impacted, these 2-inch depth dwelling nematodes, are they impacted by things like rototilling, or para-harrow is a more gentle way of incorporating amendments into the soil? Are they impacted by insecticides, amendments? And do you know anything about that, Elson or Carol?
I don't want to dominate this, Carol. You want to go first on this?
Well, I think Elson's done the science on it. I just told everybody what I knew about vineyards. I think there's some major disturbance done by tillage, which creates openings for certain pests. And if you kept some of the soil undisturbed, which you are trying to do, it's going to change things. And it'd be very interesting to see how they change.
In the case of persistent nematodes, if you moldboard plow, which you guys don't do, but if you moldboard plow, carpocapsia is now 8 inches. Carpocapsia comes back to the top 2 inches. They're moving in the profile to where they prefer, okay? In the case of tillage, you redistribute. A single plowing will move native nematodes 100 yards. We have documented that a golf course that has been treated with Feltii gets moved on the lawnmower tires of the lawnmowers. Grazing livestock in the Southwest will move them into our untreated check plots. You know, great for farmers, tough for scientists who need untreated check plots. So any soil movement moves nematodes. That's a good thing with persistent nematodes, because if you get pockets where you don't have a good distribution, the tillage, the rototilling and all of that sort of thing redistributes. So that's really a good thing for the farmers. You can see why I have gray hairs trying to do research for 30 years with these critters.
So you're saying that these tillage interventions and soil disturbance don't kill the population, don't depress the population?
No, I think they have no impact.
Great. Cool. Scott, do you have any questions for us?
You know, I'm impressed and pleased to say that a lot of what I had floating around in my head has been answered during these 2 talks, even though I came in a little bit late. Along, I think what Mike was asking, I'm always that guy that wants to just like dig into the wooded plot at the corner of the farm and see if I can do it on my own. It sounds like you're saying, you know, kind of don't waste your time. You might not get the right mix. Like I could buy from Mary or Sheilds Lab the right kit, the right species to do it. Well,
if I can interrupt you a minute. So if you buy, because they're persistent, they cannot be patented. And because you buy a small quantity and you put them on your farm, they will persist, and you can go back and you can then rear your own and spread your own. And in fact, we have documentation that's freely available on how you can rear these things.
Okay.
So you could buy a small quantity, you could treat. And in fact, as I work with my counterparts nationally in Nebraska and Texas and Iowa and all of that, my first comment is here are the nematodes. Go find a place that's undisturbed, deposit them, let them kind of adjust to Iowa or Nebraska or wherever, and then you go back to that spot to reinitiate your colonies, because I'm damn near 70 years old, you know, and I'm not gonna be around that long. So, you know, I'm trying to make everybody independent. Not only do you get adaptation, but you to your local areas, but then that becomes your source. And so that's the ideal way to do it. You know, I don't think it takes much adaptation. You're also in Jersey, are you?
Correct. I'm very close to where Mike is. Yes.
Yeah, see, I don't think that there's that much difference between the Hudson Valley climate and where you guys are.
Yeah, we're
There. Yeah,
what would be that optimal location on the farm for that repository? Is it a wooded, shady spot at the edge of the field?
grass yard. Your grass yard. Yeah, 3 widely separated patches of grass. So you know, so it takes a while for the 3 species to come together, because grass has lots of hosts. You got wireworms, you got white grubs, you got all just a patch of grass. And you tend to water it, or you tend to take care of it, where the woods is, you don't know what's going on.
Iko, I know you said you had a couple questions. Did all your questions get answered? Do you want to add those?
Yeah, I have a lot more, but just one thing that came up just in the most recent discussion. So are you saying that with the native nematodes, if you already have some existing in your soil, it's not like necessarily enough to keep your pest population down, and you need to apply like a larger application? Or is there a case where some farms naturally have these nematodes and it's, you know, enough to actually affect your pest population?
I would suggest that you have the wrong species mix.
Okay, so you need the right balance of the 3.
Well, so your farm was what, forest before, you know, it was cultivated into agriculture, right? The nematodes that were there evolved in forest. They may or may not, they're usually not adaptable because of soil profile preference to agricultural pests.
Right.
So that's where we got into the species mixes.
Okay. And I guess just one quick question. I think it came up in the chat, but I'm really interested. When you apply the nematodes, does it need to have direct contact with the soil, or can it be applied on mulches or plant residue and still be effective?
In our case, we've not looked at that. You know, we tend to be in the bigger agriculture. And so we use fertilizer stream nozzles to try and get it down to the soil surface. I mean, I know that we've worked a little bit in turf with Kyle Wickings, and there tends to be sometimes some buildup in turf, and I think they can make it through that fine. A lot depends on how much water. You know, if you can put on lots of water and flush them down through the mulch, I see less of a problem.
Thank you.
Yeah, so there were a few other questions like that about sort of how to apply these most effectively. So we covered the mulches, and yeah, so they need to have that moisture to be able to move. Really, they, as Carol mentioned, don't really move without that water layer in the soil. There was another question about, Carol, you mentioned the pressure of some sprayers that could kill the nematodes, and the screens, removing mesh screens before spraying. Have you worked with growers that use backpack mist blowers? In particular, would those damage the nematodes? Or maybe is there a way to check and see that that they survived?
Well, certainly if I was going to use a mist blower, I would try to get some nematodes and see if they have been mutilated. Remember, if you need that film of water to move through in order to get down into the soil and out of the sunlight, I think that is actually more telling and more critical the mist blower is not going to have that much water. So let's say that you kill 50% of your nematodes because they're not getting down into the soil quickly enough. I wouldn't do that unless somebody was coming right along behind me and watering, or if I was doing it in the rain.
Yeah, what about in the cases where you talked about in the greenhouse for like thrips control on the leaf?
A lot of water would still be helpful.
Mm-hmm.
I don't know anybody, direct answer to the question, I don't know anybody that uses a mist blower for applying nematodes.
Okay. Would you say that a surfactant or other type of wetting agent would help the nematodes distribute?
We really encourage that for thrips control, because on the leaves, and let's say that we want the nematode to get into the flower, you need to not have the water create little balls. If you ever watch nematodes in a water droplet, it's like sci-fi, because they are trapped. They cannot move outside of that water droplet. They are in a dome, and that dome shrinks, and they're held in that shrinking dome like a cage. They can't get out, but the surfactant would make them able to move over a wider area.
Okay.
Can I add a comment with that, please?
Yes.
So I'm working with John Sanderson, the greenhouse person at Cornell, and we've been dabbling with this for quite some time. And it really, and Carol mentioned this, it really appears that the activity of nematodes is against the pupae in the top layer of soil. So it appears that a lot of the foliar application is the runoff into the soil and killing the thrips that are in pupal form, rather than on the foliage, where you get almost instant death with UV exposure.
Yes, that's where we disagree. So the example of the 1.5 acres of flowers, there's no way they could get into the soil when it's all flower foliage in pots. There's got to be something happening. It might be happening in the flowers, which actually makes a lot of sense. The weekly treatments with low rates of nematodes is working extremely well.
Okay, there were, let's see, just a lot of questions coming in. So let's see if I can sort of lump some together. Questions about availability. So maybe I can put some links in the chat to like the IPM Labs page and other places where you can buy the commercial products, or Elson, you have a program to help growers. Well, you can sell them their initial treatment, inoculant treatment, and then they can grow their own. Is that?
Yeah, that's correct. We can provide, if somebody sends me an email, we can provide a extension publication about how to produce your own. And my earlier comment was, you find some grassy areas and you dump on what we provide, and then you can always go back to that and isolate. And we actually have a manual on how to do that.
Okay, I did put a link in the chat for that, but I think what we'll do is try to collate all the questions and answers into a document that we'll post with the video, and we can email it out to everyone who participated today, because even with the extra time, I don't know if we'll get to all the questions. Some of the others were, could you mix, what's the compatibility with either pesticides, or there was a specific question about Beauveria bassiana. Any thoughts about mixing them with other biocontrol pesticides or products?
Carol, you have an opinion?
It's mixed with Beauveria bassiana for dips quite frequently. And that would be when you're doing little plants and you want kill pests that are on those little plants. I actually believe, although I'd have to double check the scientific evidence, that Bovaria can infect nematodes. So there could be a little bit of bad interplay there, but it's used, the combination is used quite often.
So the
Interesting.
non-scientists on the call, is Bovaria milky spore? Same thing as milky spore, because that was the question. Yeah.
It's a fungus. Bovaria bassiana is a fungus that is commercialized as a product called Mycotrol or Botanigard, and probably some other names, but Bovaria bassiana kills all kinds of insect pests, including thrips, whitefly. It's used for many, many things. It's even used for housefly chicken manure.
Yeah, Carol, good point there. The question was about milky spore, which is a different organism, actually, a bacterium that folks use for Japanese beetles and other grubs. So do you know anything about interaction between that bacterium?
Milky spore is a bacterium, which is not going to harm the nematodes, and the nematodes won't harm milky spore either. I would not use the milky spore though myself. So I think the nematodes would be much better.
Interesting.
But that was for Japanese beetles specifically, was the question, yeah.
There were a couple of questions related to the temperature. So it's wanting to balance the being able to keep the nematodes moist in the soil. I often think that applying them through the drip with mulch over top so that the soil is staying more moist would be beneficial. But I'm wondering now if that, often we're using black plastic mulch, if that would get too hot under there and kill the nematodes. Certainly on a lot of our no-till farms, people are using other mulches like straw or bark and stuff like that that wouldn't have that same heating effect. But for folks using black plastic, do you think there's an interaction there?
It could definitely be a problem. Yeah, so like there's two different purposes here. One, we've got wild nematodes that are native to the area, and another, we've got nematodes that are being used like a pesticide. On either case, if that temperature, the soil is going to be above 85 for very long, the nematodes are either going to have to go down and get out of the way of that heat, or they're going to die. So it could be a problem.
Okay.
A lot of this has to do with species sort of thing. And so Heterobidites, the New York native Heterobidites are doing fine in Roswell, New Mexico for some reason under irrigation, and they're probably pushing the 85. So I think Heterobidites is a little more tolerant of the conditions than Feltii. The other issue is depending upon the strain, there's a lot difference tolerance. A student just did a thesis on the heat tolerance of all the commercial strains on a Geisman, and tolerance some up to 40 C. So what that's 103 or 4 or 5. So there's some of them some strains that are quite tolerant of the cyanonemas also. So I think it's strain specific.
Okay. Maybe related to that, there's questions about using the nematodes in high tunnels, where maybe the temperatures in the tunnels can get higher than 85 certainly, but in the soil, maybe it's okay, or.
Yeah.
Well, there's
going to be a lot cooler.
I'm sorry, go ahead.
Go ahead, Carol.
Go ahead.
It's not going to be a problem. The soil is going to be cooler than that. We run into difficulty in Florida. There's places we won't recommend nematodes.
And Elson, you had something to add there?
Well, I was just going to add that soil has a tremendous insulating ability, and the surface can be 100 degrees and you go down 2 inches and it's 20 degrees cooler. So I don't see that high tunnels would be a problem. I mean, Anna's work was, she was concerned about overheating in water before you apply it in greenhouses sort of thing, just to see what the tolerance would be, you know, and it surprised all of us that you know, 35C and 40C is not mortal, doesn't cause mortality in a lot of the commercial strains. Above that, they're dead pretty quick, but they seem to have more tolerance.
Okay. Another question I saw, are there risks to beneficial insects in the soil? And maybe this is most important with the persistent nematodes, because like you said, the commercial products are effective right away, but then they don't last for very long. But if we're talking about sort of inoculating our soil with a nematode that has broad impacts on a range of insect, well, pests, but what about beneficial insects?
Well, so nematodes are a generalist, and they will attack any susceptible insect, whether you dump them on as a biopesticide or whether they're native in the soil. I would suggest, I have not done the level of research to really answer the question intelligently, but if Steinodium carpocapsae is our most common native nematode, and it rules the top 2 inches, a lot of it's pretty hard on our beneficials as it is. You know, the carabids, a lot of carabids are in the top 2 inches. So in my opinion, I don't think we're impacting things too much. You know, we're just adding basically the lower profiles, and it becomes a kind of a conscious decision. You know, agriculture is not natural, it's artificial. So we're trying to manipulate an artificial condition with the least amount of impact. If it was natural, you know, otherwise we'd starve to death if we didn't raise food.
Yeah, there's so many combinations of nematodes with beneficial insects. So a carabid is a big beetle. We have a little beetle that we sell. It's called a rove beetle. It's tiny, and it has its own natural resistance. It's a soil predator to nematodes. So we don't worry about it so much. It actually nematodes might kill a few of these beetles, but even when Elson showed you the data on the percent of the pest that was dying, I think a lot of times it was 50%. It wasn't all of them. So a lot of even the pests have some resistance, the soil pests, against nematodes, because they're in the soil and they are naturally somewhat armored against nematodes.
It's definitely a coevolutionary arms race between the naturally occurring Bavaria fungi and nematodes. And I mean, European chafer is a perfect example. It's almost immune to nematodes. It actually has sieve plates over spherical openings to prevent nematodes from getting in. So
just evolved those naturally.
Yeah, it must have evolved in a high nematode environment. You know,
yeah, it's a good point to bring up for multiple reasons, and one being just that people shouldn't walk away from this workshop thinking that if they use these nematodes and follow the guidance that we've given today, that they'll have 100% control of all their insect pests. Because yeah, like you just mentioned, the control that we were seeing was something like 50, 60, 75% in some of these systems.
That's not quite the right interpretation. The interpretation was that in the case of black vine weevil, it took a period of 2 years to bring it from 2 larvae and two larvae per plant and half the plants infested down to non-detect. So that was a time series over time, that it takes a period of time. Yeah. And if we had gone out there with a high rate, say 1 billion nematodes per acre of commercials, we might have actually brought that infestation into control much quicker, but then you would be applying them for multiple years. So it's kind of, the problem with the farmer in that case was he let the condition get too bad, and it takes a lot longer to turn it around. You know, he basically was losing his whole 10 acres of strawberries.
Yeah. There are a lot more questions. I want to just take a pause, and if any of our CIG farmers have any questions, they want to jump back in here for a minute. Otherwise, I'll keep trying to get through as many questions as we can while we have time.
I'm good. I'd love to hear more of what is being asked in the stream. A lot of it is covering what I have to say or ask, and I've had a little time, so
Okay,
the stream.
keep plugging away. Some folks are asking about maybe sort of compatibility with vermicompost tea, whether or not you can inoculate compost with nematodes, and then that would, well, there's sort of two questions. Would it work to inoculate our compost and vermicompost bins with nematodes?
You know what, I can address that. We have people that are vermicomposters that use nematodes to control some of the little flies that get into vermicompost.
What's the second?
And they're very thankful and grateful for that. They do add some of the rove beetles, that little tiny beetle I was telling you about, into their bins as well. So they do both the nematodes and the other predators. And then as far as the compost tea goes, you know, when you're making compost tea, you let it sit for a while. I'd be a little careful about that. The nematodes drown very effectively, and I wouldn't want to kill them by drowning. Although compost tea, if it's got aeration going into it, they might survive for a while.
Let me give you a contrast to support what Carol is saying. So we have found on a huge scale that if you wash out the nematodes out of the sawdust mix and put them into a bubbling 350-gallon tank with enough nematodes to treat 150 acres, so you know you got lots of nematodes, they will actually sit there with no mortality for 2 weeks, as long as it's aerated. The minute you lose power, they're dead within 30 minutes because they suffocate. So I mean, you know, even on a huge scale, if you can have some aerators going in there, that preserves it. Another completely at the other end, we have found that you can dump nematodes in liquid dairy manure right out of a pit, and if you can spread the manure on the field within 30 minutes, you get perfect establishment. More than 30 minutes, they start dying like mad, and it's all suffocation. So liquid dairy manure provides you the UV block, so you don't have to, you can do it any time of day or night. It gives you the water to get it in. It is a god-awful mess, and I can't imagine how the nematodes can survive. But if you get it in and out within 30 minutes, you're good, you're golden. And farmers, the dairy farmers in Northern New York are loving that.
Okay, there are a bunch of questions about specific pests that we haven't talked about so far. So one was squash vine borer. Do you think any of the nematodes would be effective on those vine borers, maybe applied at the end of the growing season to the residue, or what do you think?
Let me make a general statement. I don't, and then let's go on from there. With at least with persistence and maybe with commercials, it is all about how long the insect has a soil interface. So if you get an insect that's in the soil a lot, there's lots of opportunity for nematodes to attack them. If you're talking about a very brief soil interface, the activity tends to be much less. And I'm not that familiar with squash vine borer, but I kind of think it doesn't have a very long soil interface.
I have a different take on it. So I'll just tell you what I know. So it used to be that people would literally inject nematodes into some of the vines, and it was very effective at killing. I can't imagine people going around with a syringe and injecting all their vines. So I had a contact in Minnesota at a botanical garden who started spraying their vines with the nematodes. And she claimed, and she was an IPM whiz, that it was very effective to spray. But I've never dared to make that recommendation to a commercial grower. She was spraying a few vines, and she could be very specific. But I think that they were getting inside the the holes somehow and then taking out the larvae. But I just don't feel good about it on a large scale.
Yeah. Okay, hopefully that's enough information to get started. Another question about cucumber beetles. So that's a beetle that affects cucumbers. The larvae feed on the roots. So there is a period of soil dwelling, a longer period of in-season soil dwelling larvae.
Yeah, so the nematodes are great on cucumber beetle larvae, but we're back to the same problem as with flea beetles. The cucumber beetles are coming to your squashes and your cucurbits from all around, and you want cucumber beetle control. You don't want larval control. But once again, you could do a trap crop thing where you pulled the cucumber beetles to something like Hubbard squash and killed the larvae underneath with nematodes for next year. There's so many cucumber beetles all around your farm. I don't think it's going to make a big difference.
That's a quick follow-up question. Sorry. Carol, earlier you mentioned, again, you're not totally sure this is what's happening, but you were saying in the greenhouse, it seemed like possibly the spraying the flowers was effective for surface control. And I know that, cucumber beetles spend a lot of time inside cucumber flowers. Would it not work for them because they're already at the beetle stage?
Adult beetles tend to
Yeah,
be sensitive than I think that's one of the problems.
I don't think they're quite as, they don't stay on flowers as long as thrips do. Thrips are, the thrips are in the flowers, that's their home. Whereas the cucumber beetles are going all over the place. I think the dose they would get would be low, too low. Yeah, I don't ever recommend nematodes for cucumber beetles unless people insist that they're in the greenhouse, they're producing in the greenhouse, they didn't come in from outside because they're completely screened and they want to control the larvae in the soil. Then I'll make a recommendation.
Okay. There's a question from Yoko about how often to apply the commercial nematodes. She says sometimes it says apply weekly, others twice a week. How do you know how frequently to apply?
A lot of times a single application will do it as a single application directed at the stage of the susceptible stage. There's only one time where we need the weekly treatment, and that's with the thrips. That's nowhere else. Elson, you had something to say about cucumber beetles?
No. I was trying to interject. I saw a question come up that Susan answered about Vermont. And I just wanted to give a little information. I've been working with Scott Lewins, who I saw on this Zoom, and Vic Izzo for, I don't know, Scott, if you're still there, was 5 years you've put up with me, or 6 years? We've been working very closely with them, and New York nematodes survived well in Vermont.
I think he's probably not able to unmute himself right now. Couldn't get that fast enough, but. Okay, we have another, I'm gonna just interject for a quick minute here to say that we're getting close to our allotted time. For anyone that wanted pesticide credits, we're gonna do a quiz at the end. We also have a little evaluation, so I'm gonna launch that in a few minutes before people have to start signing off. But if someone else can keep the questions going, I'm gonna try to get Noah permission to ask his question live and launch these polls.
Great. So I'll ask a couple more audience questions while we're getting Noah off mute here. Noah is our good friend of NOFA-MA and wanted to clarify his question. But I did hear, I saw a question come through about just generally wondering, do all soils have nematodes? And are there any soils that people should be particularly, prone to wanting to apply things to, like soils that have certain characteristics where they might not have nematodes?
From my experience with persistence, which range from pHs of 5 to pHs of 8, there is no problem with nematode persistence. The bigger issue is if you talk about really small pore size, so you get into the clays, things like Heterobidites cannot move around with a small pore size. That's why you tend naturally to find them in the sandier soil. And then of course you get into some of the pretty heavy clays, it's pretty tough to farm there anyway. You probably shouldn't be farming those soil. So no, we haven't come across any problem. In terms of, you know, do all soils have nematodes? There are probably some soils that don't. I mean, we do find some pockets. Sometimes the natives are really hard to find because they're in such low populations, and we get tired of, you know, bioassaying 400 or 500 samples looking. So yes, I'm sure there's some soils, but we have not seen a problem.
That's helpful. So do we have Noah able to come off mute now?
Can you hear me?
Yeah, hey Noah.
Hey, so last year I had really bad squash vine borers and cover cropped the ground where my squash field had been, and terminated the cover crop using landscape fabric. And it was really interesting when I uncovered it, there were all of these molts from the vine borer pupae all over the soil, just tons of them. And that got me thinking, there's a period of time where the soil's warmed up in the spring, like in May or early June, before the squash vine borer adults fly. And I was wondering if I applied nematodes in early May, if I might be able to knock out the population that was concentrated in my old field before they flew to my new field. And does that check out with you all? Does that seem like a reasonable approach?
Well, did the molts actually die? Did they perish? I mean,
No, it was
molts?
just protected so that I could see them, but they had gotten out and flown away. So I could see the empty shells on the surface of the soil.
Generally, pupae are more resistant to nematodes than larvae, partially because they are emitting less gases and they're less attractive. And larvae are moving around them more, so they're more exposed. So it's possible, but I wouldn't bank on it.
Thanks.
They're overwintering as pupae, so they might not be susceptible.
Yeah.
So some of that has to do with the species of nematode that happens to be there. If you're talking about an ambush, you know, they need the insect to move around. If you're talking about Heterobidites and the soil's warm, they tend to cruise all over and look. So there's some impact there. A bigger concern is just like flea beetle. You know, if you control them in your small plot, is there enough wild ones that are coming in from the fringes and everywhere else to where your effort is actually ineffective? And so that would be my bigger concern, you know, rather than effectiveness.
Yeah, so in this case, the adult is a moth that flies really well at night, so.
And it's a native insect, you know, and there's probably alternate hosts around. And I've not worked with squash vine borer, but that would be my suspicion, you know, that there's enough of a wild population elsewhere that it may not be quite effective as you want it to be.
I was reminded that we talked about the vermicompost, and Carol, you mentioned the fungus gnats and other flies. There was a question about using the nematodes to control those flies in compost. How many applications do you recommend, or what frequency of applications for controlling flies in a compost?
I am not an expert on this. I can get you an address for a guy that does this. He rehabilitates people's compost bins. But what he does is he gets it started And then he always uses some residue from an old bin to start a new bin. So some of these natural enemies go from bin to bin. So they don't use actually very much. Their nematodes are available in packets as small as 5 million. They're available 10 million, 25 million, 50 million. So you can get small quantities for that purpose.
We are at 2:45. I think we might need to leave it there with questions, Sue.
Yeah, and if Carol and Elson, if you're okay with this proposal, I'll put together a list of all the questions and my understanding of the answers that were given today. And if you guys could look it over, and then we'll post it to the group later on, that would be great.
That would be fine.
I did want to just take a quick second to give a quick plug for the upcoming NOFA-MA Winter Conference, which we have coming up. You know, we have over 30 workshops and a farmer-to-farmer section, and we do have a really strong soil health track. It's a big focus for NOFA-MAS is farmer-to-farmer education around soil health. So I dropped a link into the chat with information about our program that is coming up the first weekend of January. So we hope you can make it and join us for that. And I just wanted to say thank you so much to our presenters and to our CIG farmers and to Sue for organizing this and offering your amazing information and the great questions that were asked, and to all of you for coming. This is really, really fun. I learned so much. Sue, I'm sure you have a couple announcements and comments you'd like to make.
Yeah, just want to reiterate what Caro said. Thanks to the speakers, and I definitely learned a lot myself, so appreciate that. I did launch this little quiz for those of you interested in pesticide credits. Please just email your license number and the category to umassveg@umass.edu. And thanks to everyone for coming, and we'll see you again next time.
Thank you.
Thanks so much, everybody. That was awesome.